All right, this is the first part of the methods section for the lumber biology lab. I suggest you get the lab handout and follow along in the methods so you understand what's happening. This is the microscopic portion of the first exercise, angiosperm and gymnosperm cellular xylem structure and relationship to wood appearance and wood grain. So first, I'm going to show you some microscope slides of several different types of xylem at different magnifications. So the first one we're going to look at is corn. Corn is a monocot, right? So this is a 100x magnification of corn. This is a prepared microscope slide from a biological supply company. So what we're seeing here, remember this is a monocot, corn, so the xylem and phloem exist in vascular bundles. These are the vascular bundles. You can see the xylem, the vessel elements, along with the phloem in the vascular bundles. So if we get closer up, this is a 450x image of that same cross section. So this is a vascular bundle close up with vessel elements and phloem. So that's corn. That's a monocot, angiosperm. This is a 100x magnification of a gymnosperm, a spruce or picea. This is a cross section. So what we're seeing here, remember this is a gymnosperm, so all of these cells are tracheids. The tree is growing in this direction towards the top of the screen. So what we're seeing is late wood tracheids, see thicker cell walls, smaller opening. This is the end of a growing season, and this is the early wood of the next year. So these are early wood tracheids in 1 year, late wood tracheids from the previous year. We can also see here a resin canal, and then these are ray cells. These are files of ray cells along here. These are ray cells as well. This is the same slide at 450x, so we're just looking much closer at those late wood tracheids of 1 year and early wood tracheids of the next year. Okay, so that's our example of a gymnosperm cross section. This is a cross section of oak. Oak is an angiosperm. It's a ring porous species. So remember, ring porous species have really large early wood vessels and much smaller late wood vessels. I couldn't even fit a full ring, a full growth ring on this slide. So basically at 100x, this is the early wood cells from one growth ring going in this direction. And these are the late wood vessel elements from the previous year. So the tree's growing in this direction towards the bottom. All these other cells in between are fibers. And then these lines here going in a radial direction are files of ray cells. Okay, so early wood vessels in 1 year, late wood vessels from the previous year, fiber cells, and ray cells. I didn't do a 450x from this cross section because basically all you would see is a big early wood vessel at 450x. This cross section is of sweet gum, which is an angiosperm, but it's a diffuse porous angiosperm. So remember, diffuse porous species have similar vessel sizes across the entire growth ring. So basically all of these big white spots, those are vessels. Okay, the tree is growing probably in this direction. Basically this is the end of a growing season and the beginning is down out of the slide to the bottom. But basically you see there's very little change in vessel size across the growth ring. That tells us that this is a diffuse porous species. This is 100x in cross section. So in addition to the vessels, we see all these are fibers, and we also see rays. So on this species, I wanted you to contrast this cross-section view at 100x with a radial view at 100x. So instead of looking at the cross-section, we're looking at the radial view. And remember, I use my hand to indicate files of ray cells. So we're looking, this is a radial section, so we're looking at those files of ray cells from the side. So these cells are ray cells. Here's another file of ray cells from the side, looking at them like this. And then going up and down, these big white spaces are vessels. You can see the end of a vessel here. That's the perforation plate at the end as it connects to another vessel element below. So these big white spaces are vessel elements. And then the spaces, these red dyed spaces in between, those are the sides of fiber cells. So that's the radial section. And then the third section view is the tangential view. So again, in the tangential view, we're cutting into these big white spaces are again the vessel elements. But now because it's tangential, we're looking at those files of ray cells from the ends. So this is a file of ray cells looking at it from the end because this is a tangential cut. So ray cells, vessel elements, and then the fibers in between the vessel elements, what we're seeing in this tangential view. So those are the microscopic views. I want you to take the images that we've provided to you, provide an example of each of those as a figure, give a good caption for the figure in your lab report, label, where possible, tracheids, vessel elements, fibers, rays, early wood, and late wood in those different views in each of those figures. Now, you're not going to see all of those in all of the figures, but you need to label them where they're apparent and then describe it as described in the lab handout. So that's basically the first half of the first methods in the lab exercise.